So my talk will be about the origin and nature of nucleic acids, and I will focus on some paradoxes and puzzle pieces and a revolution in chemical evolution, what they stated there. So, yeah, let's first take a look at these images. They show a Martian landscape on the left and a landscape in Tasmania on the right. But this is just what we see on the first view. The real fun in science always comes when looking behind the curtain. So what do we see beyond that when being educated as a scientist, especially as an astrobiologist? On Mars, we see things representing high order, that's minerals, and stones as being a solid mixture of minerals, which represents a chaotic state. So we have something with high order and something with chaos. In the right, we see something in between order and chaos. We see repetitive units of certain programmed shapes based on coded information. These units, the life forms, are not just lying around over millions of years, but actually being there for the first time since a couple of years or even weeks. But being there for the first time happens repeatedly over millions of years. So as a whole, the outline remains stable. This stability achieved via replication is called dynamic kinetic stability. This type of stability is completely different to the one seen on the left on the Martian landscape. As far as we know, on Mars, we just have thermodynamic stability. If you don't mess it up with a smashing lander, impacting meteorites, or swirling dust devils, the assembly of stones and dust is happy to lie around and remain as it is, and keeping the internal structure of the stones constant. So what causes the difference between these landscapes? Basically, it's an organic molecule, but it's a very special one. We know since recent discoveries made by landers and rovers on Mars that there are very simple organic molecules in the soil. But the landscape on Earth is dominated by a certain type of molecules that, when it first appeared on Earth, made a revolution in chemical evolution. Earth, so far familiar with geology, became competent in running geobiology and biochemistry taught by these molecules. These molecules are termed nucleic acids. Yeah, in 1869, Friedrich Miescher extracted something from a cell nuclei that turned out to be something different to proteins. An organic acid rich in phosphorus and nitrogen, but without sulfur. It was about 100 years later that the sum of discoveries made since then revealed that nucleic acids are a substance with properties that went far beyond any imagination possible at the time of 1869 concerning that substance. Nucleic acids are a wonderful example of what science is about, basically. It's not only mapping and studying things in nature, such as the vast variety of molecules. It's about charting the unknown possibilities of existence. So if we came in dialogue with nucleic acids by asking them to tell us something about their very nature in the language of science and different scientific disciplines, we see nucleic acids are also messengers, having much to say about the early prebiotic conditions on Earth, the difference between self-assembly, something highly ordered, to self-organization, something with having functional complexity. Or they tell us something about the ambivalent role of water for life, or the emergence of function and purpose. They tell us something about digital information storage starting in the Archean world. They tell us something about the emergence of a new kind of stability as the very difference between life and non-life, and much more. We might still be far away from conceiving the big picture, but A closer look at some puzzle pieces we got so far from different research disciplines can already shape this view of nucleic acids, their origin and the emergence of life fundamentally. So this talk aims to shed light on some of these puzzle elements and their relations in order to give an insight into the multifaceted character of nucleic acids emergence and the interdisciplinary approach required to find insightful answers and new good questions. Now, what are nucleic acids? To give an answer is easy and difficult at the same time. This is because it depends from which point of view we look at nucleic acids and ask. From the perspective of biochemistry, the answer can easily be given by referring to the current understanding of what nucleic acids are with respect to their structure, function, and role in the intracellular network of functional biomolecules. In structural terms, nucleic acids, such as RNA and DNA, are the largest organized molecules known so far. They are natural polymers. 4 different species of a certain type of molecules, the nucleotides, act as structural building blocks for RNA and DNA, respectively. When these monomeric units polymerize into a chain of nucleotides, they form polynucleotides. Nucleic acids are examples of such polynucleotides. The nucleotide sequence in nucleic acids is of organized complexity. That means that its complexity is based on encoded information. The information that defines the sequence can be replicated, a capability of nucleic acids which is outstanding among all other known polymers. In the context of a living system, the information-coded organized complexity is functional. That means it enables nucleic acid sequences to give all information that encode the assembly of biomolecular nanomachines at the structures of cells, organs, and organisms. So, in sum, nucleic acids can be defined as biopolymers with the characteristics of organized functional complexity that has the ability to replicate. This unique combination of properties makes nucleic acids to one of the most intriguing molecules and places them within the fuzzy zone between chemistry and biology. When we change our point of view on nucleic acid and want to understand them from the perspective of prebiotic chemistry, things become weird along all central aspects of their existence. Intriguing answers given by molecular biology and biochemistry are replaced by unsolved issues in astrobiology. For example, we have these 4 nucleotide species for a given type of nucleic acids but why just 4? And why is especially AMP, the adenosine monophosphate, so common? Another issue, how to solve the water paradox? And what makes a sequence functional? And for what is all this encoding, basically? So this talk aims to point to the fascination of why it is so difficult to define what nucleic acids are when looking at them from the viewpoint of prebiotic chemistry. But exactly the understanding of this difficulty can be the starting point for young scientists from various disciplines who take up the challenge and aim to assemble the big picture of nucleic acids' prebiotic origin and the emergence of life. So let's first take a look on one of these building blocks of nucleic acids. And the thing is, why is AMP so common? If we avoid to get lost in too much biochemical details, we can recognize something very remarkable. In many very different biochemical processes, which are not directly related to each other, a specific molecule appears as a recurring element. It's adenosine monophosphate, or short AMP. This nucleotide consists of the nucleobase adenine and a pentose and 1 phosphate group. But what is so special about AMP? This issue is still open so far, but we can survey its multifaceted role in biochemistry and aiming to find properties which at least correlate with its outstanding role. When we shed light on nucleic acids in our survey, we see that AMP is one of its canonical building blocks. But there's more. AMP is also a key element in the process of gene expression in the form of terminating poly-A tails. That means it's just a sequence of multiple AMPs. Comparative genomics suggests that the addition of poly-A tails is very common in all known life. So the last universal common ancestor probably already had worked with that. Beyond these tasks, AMP has many more jobs in the molecular factory of a cell. It is involved as residues in various biomolecules, which are of central importance for metabolism and cellular signaling. But why is AMP such an all-rounder in the biochemistry of all known life? The multifunctional role of AMP suggests that it has been selected very early in the prebiotic chemical evolution towards the origin of cellular life. But what properties makes AMP so outstanding during this era? Well, there's no definitive answer so far, but we have some indications which wait to further be explored. One indication arises in experiments in which peptidyl RNAs are synthesized under conditions established for genetic copying. When RNAs and amino acids are added to this mixture, a spontaneous formation of peptidyl RNAs is reported. What is relevant here is that the reported results indicate that AMP has favorable reactive properties for the generation of a variety of biomolecules, which have a central role in cellular processes. Another indication for an outstanding property of AMP comes from our own experiments, which focus on a possibly interplay between nucleotides and nanofluidic phenomena emerging in aqueous suspensions of particles where temporal nanoconfinements of water are formed. These experiments initially aim to increase the efficiency of a certain stacking and self-assembly effect of organic molecules for nanotechnological applications, which we term organic solid-solid wetting deposition. Solid-solid wetting is something in the context of the experiments where we can form monolayers of organic semiconductors for various nanotechnological applications. In the context of our experiments, we added phosphates to one sample, and that turned out to be an efficient catalyst for solid wetting. So then we wondered what biomolecular phosphates could do and just added AMP. And AMP really surprised us with having an outstanding catalytic effect, even among other nucleotides. We see this here in our slide, as the dense aqueous suspension of particles in these experiments can be regarded as an inorganic version of the intercellular environment where densely dispersed macromolecules with nanoscale separation also generate nanofluidic effects on water between. We analyzed possible polymerization of nucleotides in such samples. And again, AMP turned out as being outstanding. These findings have prebiotic relevance. The results suggest that catalytic reactions in temporal nanoconfinements of water may have played an important part in the prebiotic chemical evolution, with AMP as a key actor. But which property can be responsible for the described outstanding catalytic role of AMP? Well, an interesting correlation appears when comparing published stacking constants of nucleoside monophosphates with the corresponding organic solid-solid wetting results. It turns out that AMP not only increases the extent of nanofluidic phenomena in these experiments more than any other used nucleoside monophosphate, but also has the highest self-stacking constant among them, and the ability to form indefinite stacks. These experiments indicate that the strange ubiquity of AMP in biochemistry could be a message from the era of prebiotic chemistry, reaching us by the conservative nature of evolution. Further research on that issue could thus help us to better develop and select possible scenarios about the origin of nucleic acids. So the next point, the next issue is, why are there just 4 nucleotide species? Why are there exactly 4 nucleotide building blocks life on Earth uses to build? Couldn't there be more? Well, there's both a yes and a no. Let's first focus on the yes. According to results from synthetic biology, there are indeed a lot more building blocks possible. The diversity of nucleic acids analogies would have been synthesized so far in order to explore the variety of structural and chemical parameters for information storage, heredity, and in vitro evolution reveals that the chemical space for synthetic genetic polymers is enormous. Nucleic acid analogues can successfully be made by replacing all subunits of nucleotides with non-natural counterparts, increasing the number of nucleotide species in a synthetic nucleic acid, or making combined modifications, as for example in the form of peptide nucleic acids. We see such a peptide nucleic acids here on the right. So all these results demonstrate that the structural scope of functional nucleic acids is huge. But for some reason, this scope is not tapped by life as we know it. And this leads us to the no as the answer. The huge structural scope of genetic polymers makes it very likely that a complex mixture of a variety of nucleic acids may have existed in the prebiotic era. As a consequence, some selection processes must have been occurred, which resulted in natural standardization and led to a few canonical versions of nucleic acids that are now used by all known life. But what could possibly have led to such a selection? Let's take a look at biochemistry. Several mechanisms have been discussed so far, which may have played a role in the selection of the homogeneous set used by life from heterogeneous mixture of genetic polymers. For example, photolysis with ultraviolet light leads to a selective impact on nucleotides. Experiments indicate that canonical nucleobases have a higher stability against photochemical reactions than non-canonical ones. There are also indications that a selective impact for superior function might have played a role in the process of natural standardization. This was derived from observations which revealed that mixed RNA and DNA oligonucleotides are of lower functionality compared to the known homogeneous one. But biochemistry is not the only relevant discipline. Complexity science has also something to add. Let's start with the genetic system. It consists of multiple diverse elements that are highly interconnected in a coordinative and cooperated way, with having the autonomy to adapt. These properties makes the system to be a complex one. Nucleic acids, as being elements of the genetic systems, are themselves complex systems. They are a manifestation of a special category of complexity, which is termed functional sequence complexity. We will see later in this talk that functional sequence complexity can only exist in a narrow section of the complexity spectrum, and that this section is nearby randomness. What is relevant for this discussion is the relation between novelty and standards within this narrow section. If a complex evolutionary system is either too sensitive or too resistant to variations and mutations, its adaptability is corrupted. This is where standardization becomes relevant. From the viewpoint of standardization, the selection of just a few versions of nucleotides from the broad chemical space may have been provided an essential contribution to balance the complexity of nucleic acids as the functional and adaptable part of the complexity spectrum. This ensures that the genetic system is neither too resistant nor too sensitive to mutations, and thus well prepared for coping with genetic innovations, for coping with genetic innovations. A comparable situation can be found in some economic systems where technical innovations with high market relevance can lead to competing technical standards defined by different suppliers. This can result in so-called format wars. They usually end when one format catches on and becomes a general standard. This leads to a much better cooperative and integrative interconnection of the new technological elements within the complex socio-economical system, thus catalyzing innovations enabled by the defined technical standards. So we have an interconnection of standardization and innovation, which might sound a bit paradox. In some recent results from different disciplines, such as synthetic biology and systems theory, support the view on the canonical set of nucleotides as being just a small fraction of a much broader chemical space available for nucleic acids. The existence of this restriction can be interpreted as an information about conditions during the prebiotic era. This message, if deciphered as and better understood, could reveal aspects on the origin of nucleic acids that were of central importance for prebiotic chemistry. The few very basic insights we achieved so far are already stunning, but there's much work ahead to get an idea of the big picture about this facet of nucleic acids' origin. So coming to the next point, what makes nucleic acids functional? To approach this issue, we can ask, where should we search for life in the universe? An obvious answer would focus on bodies where indications for liquid water exist, or in other words, follow the water. That's a good starting point, but we should be aware of a selection bias, as we would only find water-based life. So if a herd of alien sea monsters would lurk in the vast methane seas of Saturn's moon Titan, we would simply miss them. But there are other ways for searching. Focusing on metabolic byproducts, such as oxygen on Earth or methane on Mars. That's chemicals which are not stable over time on such worlds. But basic assumptions on their synthesis and stability in the complex geochemical environments may be too simple modeled. Well, another example of a more general strategy is based on thermodynamic issues and is termed follow the energy. All these approaches scout possible locations which meet our assumptions about habitability. But there's also a non-geographical location where habitable conditions exist. When we cast light on the theory of complex systems in the search for life, we find it sitting within a narrow section of a pathway or zone between order and disorder. This brings us back to nucleic acids and their origin. The outstanding character of nucleic acids among all other known molecules is the fact that they contain messages with meanings. The messages are addressed to the gene expression and regulation system. They are based on information which is encoded within the sequence of nucleic acids in a linear segregated way. If we look at the genetic code from the viewpoint of information theory, we see a unit of digital information. The codon-anticodon pairing represent a 6-bit byte of digital information. So we have information stored in nucleic acids that is linear, segregated, and digitally encoded. Well, but what does that mean for the overall nucleotide sequence? Let's have a look at complexity. The complexity of a nucleotide sequence can be described in relation to aspects such as order, algorithmic compressibility, and algorithmic function. A highly ordered sequence with a simple repeating motif, such as a poly-A tail of messenger RNA, has the lowest possible complexity. When complexity goes up, it reaches the maximum at randomness, where no order exists at all. Thus, the degree of complexity of a given sequence can be described in relation to two extrema: order, that's minimal complexity, and randomness, maximum complexity. In addition to order and disorder, the aspect of algorithmic compressibility can be related to sequence complexity. Okay, what does algorithmic compressibility mean within this context? Let's take the example of a poly-A tail of messenger RNA with the sequence A. An algorithmic compression program for this ordered, simply repetitive sequence can be very short by just reading 4A. This tells us that a sequence with low complexity has a high algorithmic compressibility. When the sequence complexity is increased, its algorithmic compressibility decreases. But there's even more about sequence complexity. It also can be related with a third aspect, algorithmic function. It is an aspect which refers to what a sequence can do itself and not what can be done for it, and thus indicates the functionality of a sequence. Functionality means, in this context, the ability of a sequence to be instructional. And this is important. When including the dimension of functionality in the description of sequence complexity, something very remarkable becomes apparent. The ability of a sequence to be instructional and thus functional turns out to only be possible within a rather narrow part of the complexity spectrum. This part appears far more near the randomness than to the ordered end of complexity and opens up a new category of sequence complexity. This category is termed functional sequence complexity. The relevant aspect for this discussion is that it enables algorithmic instructions. Outside the functional part of the complexity spectrum, sequences of various degree of complexity exist, of course, but they cannot be instructive due to a very low level of algorithmic function. So in sum, the theory of complex systems enables us to describe nucleic acids as polymers with a unique type of complexity, the functional sequence complexity. Its characteristic degree of aperiodicity opens up a window to biological organization with function. Thus, functional sequence complexity is of central importance for understanding the origin, function, and evolution of nucleic acids, and can be regarded as the outcome of non-random selection of meaningful sequences during chemical and biological evolution. As being much closer to randomness than to order, the peculiar degree of aperiodicity of functional sequence complexity enables nucleic acid sequence to be instructional and adaptive for various environmental influences and their changes. After finding nucleic acids in the narrow section of the zone between order and randomness, where algorithmic programming is possible, we can pick up this functional feature and turn to the initial issue. Where should we search for life in the universe? The characteristics of nucleic acids give us the hint to include the complexity vector in our toolbox, which allows us to search for functional complexity. This can be found in any form of a periodicity with algorithmic programming that ultimately leads to organization instead of just self-ordering, as in the case of minerals, or randomness, what's called the asphalt problem. Here in this image, we see different complexity categories. A lichen in the upper right part represents functional complexity. It lives on a rock representing nearly random complexity and is accompanied by a symmetric mineral, here the left part, which is representative of ordered complexity. The functional complexity of the lichen is located between order and randomness, but much more to the random side. Its functional complexity is made by the action of nucleic acids, which itself represents functional sequence complexity. So, coming to the next strange issue is the water paradox. Let's start with the Earth. Earth is a body in the solar system with a very low water content. Although it seems to be a water world, appearances are deceptive. The oceans are very thin varnish with respect to Earth's diameter. A census of all water, both on its surface and within the lithosphere, reveals that it is just about 0.04% of Earth's total mass. But despite water is a very rare compound on Earth, it is of central importance for the origin of life as being very toxic. It impedes the formation and weakens the stability of dissolved biopolymers such as nucleic wait. Yeah, this was too far. Okay. Being attracted by paradoxes helps a lot when we want to understand life's origin. Among the amazing zoo of paradoxes is the so-called water problem. It is one of the most nagging but also fascinating ones. The problem with water comes from the fact that it hampers the formation and stability of biopolymers, such as nucleic acid. The formation of nucleic acids is a condensation dehydration reaction that releases water. However, a product of a reaction is difficult to form when being at the same time the solvent. Thus, when occurring within an aqueous solution, a condensation dehydration reaction is highly unfavorable. As a consequence, a spontaneous water-releasing formation of nucleic acids is prohibited by water. Even if RNA has been formed under some conditions, it is spontaneously corroded by the effect of hydrolysis when being dissolved in water. We thus have an intriguing paradox. Water inhibits the synthesis of essential biopolymers such as RNA and corrodes them via hydrolysis, but at the same time is essential for running the biochemistry of all life as we know it. Several methods have been proposed so far on how to overcome the water paradox for the abiotic formation of oligonucleotides. Approaches which have produced RNA-like oligonucleotides are dealing with parameters such as temperature, chemical activation, or reducing water activity. The challenge of solving the water paradox, however, is prebiotic plausibility. Are approaches realistic? High temperatures far outside the known tolerance spectrum of terrestrial life, non-aqueous solvents, or the presence of condensing agents which are toxic even for primitive life forms are of limited prebiotic plausibility. An intriguing solution of that problem is a holistic approach based on cycles of hydration and dehydration in volcanic hot spring pools, the hot spring hypothesis that will be presented in the second session of this event today. Here I thus will just add the discussion in the scientific community of the role of minerals for prebiotic chemistry. We have a huge diversity of life that is accompanied by a huge diversity of minerals on Earth. As far as we know it, no other body in the solar system presents such an enormous variety of mineral species and morphologies. So is the presence of life and the high diversity of minerals on Earth just coincidence, or is there more? We have seen that water destabilizes biopolymers and hampers condensation reactions. Life seems not only to cope with it, but even use water as an essential element for running its complex biochemical systems. Such an aqueous interplay between destruction and construction can also be observed with respect to geology. When astrochemistry and optical dynamics work together and cast liquid water to the geology, we see a massive impact in causing vast geological changes. Water changes the geology of Earth into a highly dynamic geosystem with multiple feedback loops. At the same time, water made the geochemistry on early Earth partly to become systems chemistry and finally biology. So both geology and chemistry were told by water to become increasingly complex. When focusing on mineral surface, we see a considerable set of interactions with molecular building blocks of life. Mineral surfaces have been shown to be able to select and accumulate relevant organic molecules, raising up their thermal stabilities and enabling chiral symmetry breaking. Within our context of nucleotide polymerization, the mineral montmorillonite deserves special attention. According to the results from numerous experiments, it shows catalytic effects on the polymerization of activated nucleotides. All these approaches are focusing on the catalytic surfaces of minerals. But there's also the possibility to polymerize nucleotides into RNA with the help of mineral particles by using the interspace between particles being suspended to water. We follow this approach in our lab. Due to nanofluidic phenomena emerging with temporal nanoscopic interspaces between suspended particles, the activity of water gets reduced. And that reduces the thermodynamic barrier for the condensation reaction. This possibility to polymerize nucleotides is not restricted to a specific mineral species, as our comparative experiments with different but highly common minerals have shown. Aqueous suspension of such minerals are geologically ubiquitous. The interplay between minerals and organic molecules seems to be of significant help when solving the water paradox in prebiotic chemistry. This help from minerals might be an indication that the presence of life and the highly diverse mineralogy on Earth is probably not just a coincidence. So, coming to the next issue, why copying? What does that mean? Let's start with an introduction. When we see continents form and disassemble up again, and even mountains rise and fall over hundreds of millions of years. But nucleic acids, as one of the most fragile molecule species, persist all over the eons, despite being constantly attacked by hydrolysis, radiation, high temperature, and many others. But it's exactly this extreme fragility of nucleic acids that makes them staying there on Earth over billions of years. To comprehend this seemingly paradoxical issue and to understand how it has been solved conceptually, we have to pay a visit to one of the most influential research institutions of ancient times. The Library of Alexandria stored thousands of written documents, mainly in the form of papyrus and parchment. Environmental conditions in that geographical area, such as high humidity at the borderline of the Nile Delta, led to a comparatively vast decomposition of the rolls. To cope with it, huge effort was required to constantly replicate the library stocks. This needed a permanent source of funding. Although the museum, including its great library, was attacked several times in the course of the history, it seems that the very end came when the funding was cut. Today, we can look at this situation from the viewpoint of physics and chemistry. Information was stored via fragile biopolymers, the main constituent of parchment, as the main and the main constituent of a metastable medium, the rolls. The fragility of the medium was compensated by replication by the copyists of the museum. Replication needed a constant flux of energy, the salary, by tapping thermodynamic disequilibria. So we can say the funding. The stability of the ancient data storage device was not given due to a low chemical reactivity, but rather to another kind of stability, raised by replication. What survived as information from the ancient documents usually comes from a chain of copies of copies. By far the most amount of rolls from ancient times didn't survive over time, but their content did when having been repeatedly replicated. And this brings us back to nucleic acids. Most probably not a single genetic polymer snippet survived the billions of years since the early Archean world. But for life, this is not the point. Living systems compensate the fragility of their components and even their whole system via replication. The emergence of replicative chemistry changes everything in the prebiotic world. This is because a replication reaction is dissimilar to every other chemical reaction. It seems that replicative chemistry started with the emergence of nucleic acid species integrated in a prebiotic chemical system. The rise of replicative chemistry opened up a whole new world beyond chemical stability. It enables a molecular species such as nucleic acids to persist over time despite a constant attack of the individual entities. We thus have a new kind of stability, which is termed dynamic kinetic stability. What is the characteristics of dynamic kinetic stability? A system based on dynamic kinetic stability remains in a steady state as long as it is at its replication rate is equally or higher than the decay rate. It is opposite to thermodynamic or chemical stability, but includes it at the same time. A chemically metastable system has reached a local minimum of Gibbs free energy. This can happen spontaneously via a downhill reaction by the release of energy. Such a system remains stable as long as there's no intake of energy. In contrast, a system with dynamic kinetic stability needs to be continuously reactive via an intake of energy, the metabolism, in order to maintain replication. Its elements have to be of low chemical stability in order to overall maintain dynamic kinetic stability. So DKS has many fundamental implications for prebiotic chemistry and biology. What is important for our discussion is the consequence of dynamic kinetic stability for nucleic acids. It makes this molecular species with replication capability extremely stable over billions of years, despite or even because of an extremely low chemical stability. So let's sum up. In someone viewed for a broader perspective, nucleic acids seem to be optimized by prebiotic chemical and biological evolution to work with very different types of stability at the same time. Thermodynamic stability with respect to their building blocks and their interaction to gain long-term genetic information storage. Functional stability and the stability of being adaptable, both maintained by functional sequence complexity. And we have dynamic kinetic stability maintained by the ability to replicate. It's very easy to run into forming minerals with high order or asphaltization, randomness. But how prebiotic chemical evolution managed to reach and combine these various and highly different kinds of stability in one molecular entity, the nucleic acids, and balance them at the narrow peak of functional complexity could only be revealed by achieving an even bigger picture. But this needs more puzzle elements, and this waits for being discovered and collected by those who take the challenge. So thank you for your attention.